And once it was proved, then the next task was to establish a relationship between the DNA and the protein that is encoded. That means uh, which is going to execute the information that is there in the DNA. So, although it was proved that the DNA is the genetic material, there wasn't enough evidence in order to say this that protein is related with DNA somehow, and the function that is attributed to the protein is actually coming from, that means the information for that function is coming from the DNA. So, considering that, there were a few other studies that were conducted. First, evidence actually was obtained much before the discovery of DNA as the genetic material, as we saw it in the last class itself. It was uh, it was on the basis of the studies that were conducted by Archibald Garrett and his supposition about association of DNA with protein came up from the studies that were conducted by Archibald Garrett and association of a disease which is called as phenylketonuria. Uh, what actually happens, alkaptonuria, and what actually happens during alkaptonuria was was a peculiar condition that homogentic acid accumulates and it is excreted in urine that gives the urine of the patient a characteristic black color. And that's a hallmark of alkaptonuria. And considering the occurrence of this particular disease is because of that means is under hereditary influence, the occurrence of this disease, in the sense that if if the parents are affected, then there is always a situation that the offspring will be affected. Considering this as the basis, it was established by Archibald Garrett's observation, on the basis of observations done by Archibald Garrett, that there must be some connection between the hereditary unit or the genes, as we call it today, and the condition that is alkaptonuria. Later on, however, there was a definite evidence that was obtained in this direction on the basis of the studies. These were very elaborate studies that were conducted by Beadle and Tatum, and that was on Neurospora crassa. And we saw this in the last class itself, that how it was proved on the basis of mutagenic radiations, that when there is a mutation affecting the DNA, then that mutation is relayed onto the protein, and thereby the activity associated with that protein is affected. So, although Beeden and Tatum at their standpoint gave only the relation between gene and the enzyme, later on, however, it was known that it's not a single what you can say polypeptide that is making up the protein. There could be more than one polypeptides, and each polypeptide is encoded by a different gene. So when different genes come together, then there could be a possibility that the enzyme function is under the control of different genes brought together. So considering this, the hypothesis that was given or established by Beadle and Tatum on the basis of their studies was actually upgraded, and that was later on called as one gene, one polypeptide hypothesis, rather than calling it as one gene, one enzyme hypothesis. Now, this relies on to the very basic fundamental understanding about mm, the central dogma of molecular biology, as we saw this in the last class as well. That when we are talking about the relay of genetic information, then there is a gene uh, that actually harbors the information, which is transcribed through a process of transcription. That's what we are going to study in greater detail into a molecule, which is messenger RNA usually, and then it is further translated and converted into the protein execution based information. Now, this is the general scheme. However, when we say gene expression, it is not only limited that it will it will only encode for a protein. Sometimes it stops here. So that is one part. But before we go into that and analyze it in greater detail, there are a few things which must come to your mind right in the beginning. First, when you look at this scheme, do you feel that this is a good scheme to go along with, or do you feel that there is some problem in this scheme, and that problem needs to be solved in order to make this scheme better suited for all the organisms? Yes, anybody? Okay, what you can do is that in case you think that this scheme is good enough, and this, say, time tested kind of a scheme of relaying the genetic information, then you can raise your hands and let me know that, okay, how many of you are saying that this scheme is good enough, there is no flaw in the scheme? Yes. So none of you actually
That
seems.
everything is okay in this.
Okay, Janvi says everything is okay. Bhavika is with Janvi saying that the scheme is good enough. Okay, one vote coming your way. Janvi, that's it. What about others? Do you think that this scheme is good enough, or there is some problem in this scheme? Okay, let us understand it like this now. Fine. There are a few things that one must consider when we are talking about this kind of relay of genetic information. First and the foremost, before we go and describe all that, and we talk about that how this scheme is suitable and how it is not suitable, and we discuss it out, let me first ask you this question: can anybody amongst you, can any one of you define for me what is a gene? So, anyone? I am not looking for the bookish definition of the gene. I'm just trying to find out that can you make out a difference between a segment of the DNA and the gene? Because we are studying molecular biology here. So I'm not interested in the genetic definition of gene. That means it is a hereditary unit. I'm not interested in that definition. I want a better definition describing molecular wise, or under molecular terms, that what do you think will act as a gene? Yes, anyone? It is very essential in order to understand the later course of our transcription studies. So will be good if anybody gives me a proper definition of the gene. Akanksha says that entire DNA sequence required for synthesis of a functional protein or an RNA molecule. Okay, so if I give you, Akanksha, this definition is good enough, but let me give you a perspective. If I give you a segment of the DNA, say this is the DNA segment I am giving you, can you tell me that whether this will qualify as a gene or not? Yes. So there seems to be some response. Janvi says no. Akanksha also says no. Okay, if you know this definition, Akanksha, if you are putting this definition of gene, that the DNA sequence which is required for synthesis of functional protein or RNA, then you should be able to tell me that whether this DNA sequence qualifies as a gene or not, right? Okay. So what do you look in the DNA sequence if you want it to be acting as a gene? Okay, did this question appear in your mind previously anywhere? That if I give you, say, a random DNA sequence, can you make out, looking at that sequence, can you make out that whether it will qualify as a gene or not? So there has to be something special in the DNA sequence that will make it act as a gene, right? Yes or no? Okay.
Sir, we need a coding gene.
You need a coding gene. Okay. How do you make out that whether it will be a coding sequence or will not be a coding sequence? I'm saying just looking at the sequence, you should be able to find out that whether it qualifies as a gene or not. Why this is important? First of all, why do you think that this distinguishing feature is important? Yes. Anyone? Okay. Do you think that this is an important question that should be asked? Or do you feel that this is okay, we can give it a pass? It's not necessary that we should answer this question. Okay, Janvi says that it is yes. I don't know for what she has said yes, whether she believes that this is an important question to be asked or not. Okay, it's an important question, says Janvi. What about others? Can we go ahead without answering this question? Okay, Akanksha and Janvi say that it's an important question to be dealt with, right? So, so you should know the answer, right? Because you have been studying gene, I don't know from which class?
11th.
11th class. Okay, good. So from that point onwards, it's surprising that you never asked this question to yourself. That when I give you a sequence of DNA, even the crude possible definition of the gene is that it is a segment of DNA. It's a very crude definition. But not every segment of the DNA can qualify as a gene, right? So there has to be something special added to that DNA sequence that will make it qualify as a gene. So what special features do you look for in the DNA sequence so that you can find out that whether this can act as a gene or will not act as a gene? One important, yes, Bhavika, go ahead.
A promoter region where transcription, where the transcribe the molecules that help in transcribing the segment of gene can sit and stabilize so that they can start the transcription mechanism.
Okay, so Bhavika says now I'm putting the points together which are coming my way. First point was given by Janvi, sorry. Janvi said that it should be a coding sequence. It should qualify as a coding sequence. So how do you look at that? That's it. Perhaps in that direction, Bhavika has given us some clues. Bhavika says that there should be some region which is normally referred to as promoter site. We will talk about this later. Or for simplicity to understand, we can consider this as transcription start site. Good enough, Bhavika? If I say this?
Okay, but just after that site transcription starts, the site where the molecules sit.
Yes,
That
technically yes, but it is the mark from where the transcription actually begins. We will talk about this in greater detail. But technically, yes, that the coding sequence or the transcription start site is slightly different from the promoter region. Good enough. But for present understanding, let us consider that I can talk about it as a promoter sequence or the transcription start site from where the the process actually begins, right? Anything else you would like to have in the DNA sequence that will make it qualify as a gene apart from these two requirements: one, coding sequence, and two, promoter sequence from where the transcription begins, because transcription is the process which will be involved in the synthesis of the product of the genetic information. Yes, anything else you would like to have in the DNA sequence that will qualify it as a gene? Okay, let me give you a hint. Maybe you will find out the answer. Say suppose this is your DNA sequence. So what Janvi says that there should be a coding sequence. Now there is no way by the way of which you can actually find out that whether it is a coding sequence or not. Okay, but if there is just some sequence which is there from the promoter site, ahead of the promoter site. So if you can find out because there is a definite sequence that can be called as a promoter. We'll talk about this later on. So if there is a promoter and ahead of it there is some sequence, then perhaps you can consider that as a coding sequence. That is one part. Okay. What else you will want? So at the promoter sits the transcription machinery, which is basically steered by RNA polymerase. And then there is a coding sequence. What else you would want from a gene?
something
Yeah.
something that can terminate the transcription at a point.
Yes, essentially, if you can begin from a particular point, you should also be able to find out that where you should stop, right? That is called as the termination site. So these all sequences have to be there in the DNA on the basis of which you can consider that segment of DNA. If I give you an arbitrary segment now, then will you be able to make out with that whether it is a gene or not? Yes. If you look at the promoter sequence, if there is a promoter sequence in that segment, then you know that some sequence ahead of that must be the coding sequence. And apart from it, if you find out that there is some site which looks like a terminator also in this entire sequence of the DNA, then perhaps you can find out and say this, that okay, this entire segment of the DNA can be called as a gene. Good enough for everyone? Yes? Or do you want anything else to be added over here, apart from all this that we have talked about? Essentially, these are the three portions that should be there. One, promoter; two, the coding sequence; and three, the termination site. If all these are there as a part of the DNA sequence that has been provided to you, then and only then that DNA sequence can qualify as a gene. If any one is missing, then perhaps that will not qualify as a gene. Good enough? Should we go ahead from this point? Okay, fine. Now there are a few more things here. Although we say coding sequence is the one which is going to be responsible for harboring the message that has to be relayed for further execution, these coding sequences can be of two categories broadly you can consider. One is the coding sequence will be responsible for encoding a messenger RNA molecule, which can be further converted into protein. Or else the coding sequence can also be that sequence which is responsible for synthesis of other non-coding RNAs. Non-coding RNAs are transfer RNAs and ribosomal RNAs. Do you agree with me? Can this gene be still called as a gene if it is encoding for transfer RNA or ribosomal RNA? Or will you call it as a gene only if it is responsible for synthesis of messenger RNA? Question is clear?
Yes, sir. Yes, so we can call this
Okay,
segment
so
gene.
you can still call it as a segment of gene, right? Or you can still call it as a gene, because here the gene expression doesn't only mean protein expression. One thing that should be clear in your mind. So transcription will happen for all those genes which are coding for mRNA as well as for transfer RNA and ribosomal RNA, right? Most of the genes actually encode for transfer RNA and ribosomal RNA. Only a few genes are responsible for encoding proteins. So one should be very clear in their mind that gene expression can stop here. If the product is transfer RNA or ribosomal RNA, right? It will not go for translation, correct? Translation will happen only if the gene is encoding for a protein. Fine? Yes, should we go ahead from this point? Or there is anything that needs to be clarified further? Okay. So when we say now that transcription is itself a process. That means you can consider this as one of the most important aspects of gene expression. So studying transcription makes the things more interesting. Yes, Akansha has something to say? No, okay, fine. Now there are a few other aspects that we need to look upon to. Okay, one aspect is that which of the two strands of the DNA, because DNA consists of two strands, right? Which of the two strands of the DNA will be used for synthesis of a complementary RNA molecule? I am not saying messenger RNA, okay? I am saying RNA because the product of gene expression is actually RNA. Okay. Now, whether that will be responsible for synthesis of protein, that's a different story. But the product of every gene expression is essentially an RNA molecule. Do you agree with me? Yes. Do you agree that the product of every gene expression is essentially an RNA molecule? And that RNA may or may not be used for protein synthesis. Is it good? Yeah. Okay. So
Yes, sir.
so now we need to focus on to the DNA sequence and need to find out that which of the two strands of the DNA will be utilized for RNA synthesis. And here the terminology related to gene expression comes into play. That will be used all throughout our discussion of transcription in all the classes, that subsequent classes that we are going to study transcription in. So simple nomenclature goes like that: the strand which is complementary to the sequence of the RNA product, that strand is called as a template strand. This is a simple nomenclature. Okay, the strand of the DNA or the gene which is complementary to the sequence of the RNA product is referred to as a template strand. Is it good enough?
Yes, sir.
Okay, and the other strand which is actually complementary to the template strand in the DNA itself, that is a non-template strand. Right? This is a simple nomenclature. Now there is another nomenclature coming over here. Okay, one strand is actually called as a sense strand, while the other strand is called as an anti-sense strand. Okay, so one strand is called as a sense strand, while the other is called as an anti-sense strand. Now, have you ever heard about this nomenclature previously?
Yes, sir.
Yes. So can you tell me which of the two strands here, which are named like template and non-template strand, which of the two will be called as a sense strand and which will be called as an anti-sense strand? Yes, anyone?
Antisense will be template.
Antisense will be the template strand. That's what Janvi says. How many of you agree? Yes. Janvi says that antisense strand is nothing but the template strand. How many of you agree? Okay, Bhavika says that the template strand is the sense strand. How many of you agree? The reason I have asked you this question is just to bridge the gap that you are having in your understanding here. Okay, now, okay, Janvi, would you be interested in clarifying that what Bhavika is saying is correct or what you are saying is correct?
Sir, antisense is a non-coding.
Antisense is the non-coding strand. Okay. So, but does that mean that it is not it is a template strand? If it is non-coding, does that make it a coding, make it a template strand or an anti-sensor strand? There are several names for the same, right? So we should be able to find out that what means what. Bhavika, would you like to clarify that what you said is correct and what Janvi is saying is incorrect? Or
Sir, I think that the sense and anti-sense nomenclature is because of the directionality in which the transcription machinery can sit and start transcribing. So the
yes.
template strand, which is coding uh,
Hmm.
or mRNA,
Hmm.
its directionality will be called as the sense, and the other one will be called as anti-sense. That's what I think.
Okay. So you believe that the template strand will be the coding strand. That's what you want to say, Bhavika?
The template strand is I am a bit confused in coding and non-coding
Okay.
nomenclature.
Okay. Sarvesh has something to say
Same.
here. Yeah. Sarvesh has something to say here. Sarvesh says that non-template strand is coding strand and is also called as an anti-sense strand. So non-template strand is anti-sense strand. That's what you are trying to say, Sarvesh? Janvi is again confused. So, okay. So let's solve this riddle. This most of the times students are not very clear about it. Okay. Now just imagine I have colored these two strands as just to distinguish them. Okay. Now if you look at this sequence of the non-template strand, it is same except that the T's are replaced by U's here. Okay. So the non-template strand is essentially, okay, Bhavika says that coding strand is the one which has similar code as that of mRNA. Okay. So now perhaps Bhavika is coming online. Okay, fine. So basically, I'm trying to explain the same thing to you so that there is no confusion in your mind from this point onwards. Okay, first thing first is that if you look at this sequence of the non-template strand, it is exactly same as the RNA product, right? Except that the T's are replaced by U's in the RNA. That is usual, correct? So when you look at this sequence of non-template strand, it makes sense to you, correct? It makes sense? Yes or no?
Yes, sir.
Okay, so the non-template strand is actually the sense strand, which make by reading which you can make out some sense, right? However, if you read the template strand, it will be difficult for you to make out the sense because it is written in reverse order, correct? Or complementary order, if I say like that. So it becomes an anti-sense strand. Good enough? Yes or no?
Yes, sir.
Okay. In the similar way, if I look at the sequence which is of the sense strand, it makes sense to me and it can be considered as the coding sequence. Okay. And obviously the template strand becomes a non-coding sequence then. Okay. Other way around, there is another nomenclature that perhaps many of you might not have come across. The non-template or the sense strand is also called as the positive strand. Okay, and the other one, that is the template strand, is also called as the negative strand. So all these nomenclatures are used frequently in different texts, in different literatures. But when they are stated, you should be very clear in your mind that what exactly is the meaning of these nomenclatures. Is this fine with everyone now? Any confusions?
Sir, one question.
Yes, go ahead.
Just because it's in uh, coding gene, that's
Mhm.
what it is called as positive.
No, see, one thing you need to understand that when you are reading a gene sequence, then you are actually interested only in the coding portion of the gene, right? And if you want to read it properly, then you should be able to make sense out of it. So unless you read the sense strand, it will not make any sense to you. Am I clear enough?
Yes, sir.
Okay. So when we say that we are looking at the coding sequence, then we are actually looking at the sense strand. We are not looking at the anti-sense strand. Okay. And one more thing that I need to make it clear to everyone, the anti-sense strand is not called as a non-sense strand. Okay. Because many of the students have this again, that means we have come across this kind of a mistake that the students normally commit. Opposite of sense is nonsense, and they start writing nonsense strand. Okay, it's not a nonsense strand. It is called as an anti-sense strand, which means that it is complementary to the sense strand. Is that fine enough? Should we go ahead from this point? Is the nomenclature thing pretty clear in everybody's mind? Okay, so let's move ahead from this point onwards. Bhavika, if you are having any confusion still, or is it fine? Should we go ahead from this point?
Yes, sir, we can go ahead.
Okay, fine. So, so now the big question that appears, okay, and that should appear in your mind as well. Once all these fundamentals are clear about sense strand, coding strand, anti-sense strand, template strand, and all that stuff, because these terminologies we are going to use next when we study transcription in greater detail. But one question is a bigger one, and then that needs to be addressed. Is there any need of transcription? Do you think that there is a need of transcription reaction? Yes. Do you feel that there is a need? Survey says yes. See, there is one fundamental question that I am putting up in front of you. First and the foremost, out of the two, RNA, which is the product of transcription, and the DNA, which is the information bearer. Okay, the information is there in the DNA. We are converting that information into an RNA information, right? Do you think that there is a need of this process? Do you think that it is a good way of relaying the information? So say suppose your purpose is to synthesize protein. Okay, now we are looking at only one purpose. The purpose is to synthesize protein. Okay, which do you think is a better option? Converting the information that is there in the DNA directly into protein, or converting that information into some transient molecule first, which is RNA, and then translating that transient molecule into protein?
Making an RNA first,
Why do you think that that is important?
so that we can control the protein synthesis whenever to just whenever required?
Whenever required, okay.
Like,
You will synthesize RNA first, so you will spend energy in RNA synthesis first, right? And then there will be a machinery that will decode the RNA, and then you will get the protein synthesized, correct? So if I want the protein, if I want the information to be executed immediately, can I do that?
if there is an
Sir,
immediate
please repeat.
Yes.
Yeah, okay, Bhavika, continue.
Sir was saying that if we can we just can we instantly produce protein, like
Yes.
in 2 steps? Yeah, if there is a need of protein in the body, then then making protein instantly by the intermediate step also makes sense, because when there is not a need of protein in the body, and if there is a
Is there any such situation when there is no need of protein in the body?
No, we need it.
Yes. So now my question is simple. I am putting it in slightly different manner now. Just imagine this. If I want some process to happen, okay, that means is it good that I directly talk to the person who is going to do that process, or is it good that I tell somebody and then that somebody tells the person to execute the process? Yes. Did you get the point? So say suppose a principal madam wants you people to fill in the examination forms. Is it good that principal madam directly tells you that you fill in the examination form, or is it good that she wants or she tells us first and then we tell the students that you need to fill in the examination form? What's good for you?
Second option.
Second option. Why so? See, it may happen like this, that principal madam tells me that you should inform students that you should fill in that they should fill in the examination forms, and I forget telling you, right? Who will be affected then? Will the process happen?
Students.
Obviously. Will the process happen?
No.
No. So is it good if it is coming through me, or is it good that it directly comes from the principal to you?
But it's not her responsibility.
Who told you that? If it is
It
not,
might be going step by step, right?
no, but if you have steps in between, is there a chance of some mishap happening in between?
Yeah, there are chances.
Yes, exactly, there are. So if in the same manner, if the information is there in the DNA, why can't I directly convert that information into protein? Why do I need an intermediate molecule, right? And if I compare the stability of the two, which is more stable, DNA or RNA? Come on, according to you, which is the more stable molecule, DNA or RNA?
DNA.
Obviously DNA. That's why you have chosen it as the information bearer, right? Because you can store the information there with high stability. So is it logical to have your stable information getting converted into some unstable information, and then that unstable information being relayed and being converted into someone who is going to execute that information? Yes.
So not getting.
What you are not getting? Just imagine this now, okay? ये सब लोगों ने स्कूल में किया होगा. Meaning you stand in a line, and the first person is told something about a particular story, and then that person is supposed to relay that story through intermediate people, and the last person standing in the line is then asked for that what was told to the first person. The information when it comes to the last person, is it the same information that is given to the first person?
No.
No. What does that mean? It
Half
means that when you are passing the information through intermediate steps, at every step there is a chance that the information will be slightly changed or obscured, right? So when it is coming to the execution stage, that execution might get affected, right? Yes or no?
Yes, sir.
Yes,
Yes.
sir.
So does that seem logical to have an intermediate molecule in between?
No.
No. So why do you have an intermediate molecule in between then? Is this a important question to be addressed? Do you believe that this is an important question? Okay. So can you find out an answer for me? What I will do today is that I will give you this entire day to think over it. Okay. And then tomorrow we will